Hello folks. This voiceover focuses on several examples of post-transcriptional regulation of gene expression. We've seen the control of gene expression at the level of transcription, specifically the level of assembly of the transcription initiation complex. Here we'll look at how cells control the amounts of protein they make by regulating events after transcription initiation. The first example we'll consider is that of small interfering or siRNA. In some ways, this is an unusual and exceptional example because it's part of a eukaryotic cell's defense mechanism against invasion by viruses and transposons or transposable elements. At some point, these invaders produce double-stranded RNAs. Selective double-stranded RNA-specific ribonucleases can detect the double-stranded RNA and hydrolyze it down to smaller fragments. Some of the smaller foreign RNA fragments can hydrogen bond to complementary regions of the larger invasive RNAs and target them for degradation. So here are the specifics. Once made in the cell, the double-stranded RNA is recognized as foreign by a nuclease called dicer, the lavender-colored objects here. The resulting fragments, somewhere between 20 and 25 nucleotide pairs long, are the siRNAs. The siRNAs become single-stranded as they combine with RISC proteins that are normally made by the cell. This forms a RISC or RNA-induced silencing complex. So RISC is a ribonucleoprotein complex, isn't it? Remember that ribosomes themselves are ribonucleoprotein complexes, so the combination of RNAs and proteins into a useful structure in the cell is hardly unknown. The single-stranded RNA in the RISC can H-bond to one strand of the foreign double-stranded RNA and catalyze its rapid hydrolysis. The hydrolytic activity is a property of one of the RISC proteins. The next example is microRNA, or miRNA for short. miRNAs are also short double-stranded RNA fragments that target other RNAs for destruction. But unlike miRNAs, they're not foreign molecules. Rather, they participate in the normal control of expression of cellular RNAs. They regulate the rate of mRNA turnover. miRNAs are actually transcribed from miRNA genes. These genes, like those for ribosomal RNA or transfer RNA, do not encode proteins. After transcription, the long RNA forms a secondary or even tertiary structure by internal H-bonding. In the nucleus, or during transport to the cytoplasm, the RNA is catalytically hydrolyzed into short fragments, the miRNAs, which then act in the cytoplasm. Here in the next step, miRNAs bind to RISC proteins. The RNA in the RISC is single-stranded and can complement or hydrogen bond more or less well with cellular RNAs, as shown here. If the match is good, the target mRNA is rapidly degraded using the RISC nuclease. If the match is weaker, the hydrolysis of the messenger RNA occurs, but more slowly, and uses other nucleases not part of the RISC complex. Both si and miRNAs are being looked at as possible chemotherapeutic agents. If we can get them into specific cells that are overexpressing, say, cancer genes, these small RNAs could selectively block their translation by destroying them. Let's take a look at another mode of regulation. Riboswitches are a bacterial phenomena. Riboswitches form when the 5' ends of messenger RNAs fold over to form quite specific secondary and tertiary structures as they emerge behind the RNA polymerase during transcription. In this example, the mRNA shown is in the pathway for purine biosynthesis. If cellular concentrations of guanine, a purine base, are low, the gene is actively transcribed. But if guanine concentrations are high, the cell requires less guanine. Then the excess guanine recognizes and binds to the stem loop structure at the 5' end of the mRNA and the riboswitch region undergoes a conformational change, forming a new stem loop structure that looks and functions a lot like the transcription termination sequences that we've seen normally form at the 3' end of many prokaryotic RNAs. The result is, as you see, the dissociation of the RNA polymerase from the DNA and a premature termination of transcription. You can think of this mechanism of regulation as being at the level of bacterial transcriptional elongation. Okay, we've looked at two newly discovered mechanisms for regulating the amount of a specific mRNA in a cell by controlling its turnover rate, and one mechanism of control of transcriptional elongation. In the final two examples, we'll consider how cells use small metabolites, or even proteins, as signals of their biochemical status to regulate gene expression by controlling translation itself. The first example is one that's been known for quite some time, involving the coordinated synthesis of hemin, or heme, and globin, the two components of hemoglobin required for oxygen binding. As a reminder, recall that vertebrate hemoglobin has a quaternary structure consisting of 2 alpha and 2 beta polypeptides. Each of the polypeptides, when folded, binds a heme group. In humans, globins and heme, or hemin, are synthesized in reticulocytes, produced by bone marrow. When the reticulocytes mature, you may remember that they lose their nuclei and become erythrocytes, or red blood cells. The erythrocytes no longer make either heme or globin. Heme and globin synthesis then in reticulocytes are coordinated, so the cell does not waste energy making too many of either one or the other globin chains or making too much heme. Here's the regulatory mechanism in some detail. Translation initiation factors in both prokaryotes and eukaryotes enable the assembly of the initiation complex, which consists of a messenger RNA molecule, the small ribosomal subunit, and several initiation factors. In eukaryotes, this process involves initiation factor 2, or EIF2, which is similar in function to the bacterial IF2. First, EIF2 binds GTP and an initiator methionine tRNA. Then EIF2 and several other eukaryotic initiation factors, or EIFs, deliver the initiator methionine tRNA to the messenger RNA during the assembly of the initiation complex. In the next step, the large ribosomal subunit binds to the initiation complex, after which the GTP is hydrolyzed, still bound to EIF2, and all of the initiation factors come off. If translation is to continue, the GDP on EIF2 must be replaced with another GTP. This is facilitated by other initiation factors not shown here. If there's no need for further translation of globin polypeptides, an enzyme called HCR kinase catalyzes the phosphorylation of EIF2 shown here. On the other hand, if hemin levels are high in the cell, which would signal a need to make more hemoglobin, excess hemin, as shown here, can bind to HCR kinase at an allosteric site. The resulting conformational change then blocks the kinase activity so that, in fact, GTP can replace GDP on EIF2, and globin mRNA translation can continue. Look back at the bacterial riboswitch regulation of gene expression, in which 3D structural folding of the 5' end of the bacterial mRNAs can interact with metabolic regulators of transcription. In this next example, the last one, the 5' end structures of some bacterial mRNAs interact with small metabolites or regulatory proteins to control translation initiation. In the absence of the small metabolic regulator or protein, the ribosome binding site acts efficiently to allow assembly of an initiation complex and the binding of the large ribosomal subunit. At this point, the ribosome quickly recognizes the initiation codon, AUG for methionine, and then the bacterial mRNA is efficiently translated. Regulatory small metabolites, or regulatory proteins, if present in sufficient concentration, recognize and bind to the 5' end structure of the messenger RNA that is trying to be translated. If they bind, assembly of the initiation complex is prevented, or the initiation complex together with its large ribosomal subunit, is broken down, aborting translation. To summarize, until recently, control of gene expression was thought to be a matter of cells controlling the initiation of transcription. Most regulation was thought to be by transcription factors binding to regulatory DNA sequences, such as enhancers. The forms of gene regulation shown in this presentation reveal that gene expression can be controlled at many points between the synthesis of the message, its translation to a polypeptide, and the longevity of the messenger RNA in the cytoplasm. One mechanism, involving small interfering RNA or siRNA action, seems to be a novelty, in which cellular ribonucleases evolved to regulate normal mRNA turnover, the RISC proteins, were co-opted to recognize and destroy the RNAs of invading viruses or transposons. And that brings us to the end of this presentation.